DNA is a huge strand of 4 similar building blocks that make up your unique genetic code. This code tells all the cells in your body what to do, like a computer code that tells a computer what to do. But like the glitches that sometimes develop in computer code, changes in DNA called mutations can occur. They're often harmless, but some can create problems. Certain mutations passed down from your parents can put you or your blood relatives at greater risk for disease. They can also affect your body's response to medicine and chemotherapy. As you grow and the cells in your body routinely multiply, other mutations can happen due to your environment, diet, habits, or just plain chance. If these mutations occur in the wrong places, they can send faulty signals to your cells. This may cause cancer or other diseases to develop. Understanding your unique genetic code and its mutations is an important tool for your doctors. Here at Moffitt, it helps them customize your care. Genetic testing takes a sample of your blood, tissue, or tumor and isolates your DNA to read the code. This provides clues about which medicines work for you, how a tumor might respond to certain therapy, or how aggressive a disease could be. These details help doctors tailor your best prevention or treatment plan. Think of it like buying a suit. You might be okay with one bought off the rack, but it's not quite like having one made for you to fit just right. Genetic testing is safe and accurate, but the results could be uncertain or maybe even something you didn't expect, like your risk for a certain disease.
Circulating cell-free DNA is fragmented DNA released into the bloodstream following cellular apoptosis or necrosis, and can be found in low levels in plasma in healthy individuals, as well as in other bodily fluids such as saliva, cerebrospinal fluid, and urine. Patients with cancer tend to have higher levels of circulating cell-free DNA, owing to the release of DNA from tumor cells into the bloodstream. Cell-free DNA released from tumor cells is referred to as circulating tumor DNA, or ctDNA, and is typically obtained and detected from peripheral blood samples. Because of the short half-life of ctDNA, which is approximately 1 hour, and the possible range in the amount of DNA fragments released by tumors, the proportion of cell-free DNA that is made up of ctDNA can vary greatly, from less than 0.1% to more than 90%. Sensitive detection methods, such as mutation-specific PCR-based techniques and next-generation sequencing, are used for ctDNA analysis to optimize the detection of point mutations in ctDNA, even when the fraction of ctDNA is small. At present, the analysis of ctDNA has many research applications and only a limited role in clinical oncology practice, but it holds the possibility for advances in the areas of cancer detection, treatment, and management. ctDNA analysis has potential for use as a surrogate or adjunct to standard of care tumor biopsy testing to diagnose cancer and perform tumor molecular profiling, particularly for patients in whom a standard tumor biopsy would be difficult or unsafe. ctDNA could play a role in tracking therapeutic response to cancer treatments, since ctDNA levels in individual patients correlate well with changes in tumor burden and treatment response. The short half-life of ctDNA is a potential advantage in measuring real-time tumor burden as compared with standard tumor markers, which have half-lives of days to weeks. ctDNA analysis could be used to monitor for possible acquired resistance through tumor subclones that emerge under the selective pressure of cancer therapy. ctDNA could be analyzed for the early detection of new cancers or relapse in patients after surgery or adjuvant therapy. And finally, ctDNA analysis shows potential as a screening tool for early cancer detection in asymptomatic patients. Cell-free DNA analysis is an emerging technology with potential clinical applications in oncology. Effective clinical integration will require further prospective study, as well as an understanding of its advantages and limitations.
A simple routine blood test can diagnose a serious disease. Blood work tells you not only the source of the problem, but its progress. A defined treatment plan is then matched to the diagnosis. Can healthcare go further? In 2009, there was great excitement over circulating tumor cells that could be found within the blood and determine progression of the disease. Could there be a test that goes deeper than the cell? The next step has arrived. The liquid biopsy seeks signs of actual DNA. Cell-free circulating tumor DNA, ctDNA, is shed from a tumor into the bloodstream. ctDNA is greater than 100 times more abundant in the blood than tumor cells. The frontier of the liquid biopsy is wide open. Experts believe it's only a matter of time before catching and treating cancer is as routine as your annual checkup.